Hello, everybody. Today we're going to look at hormones and metabolism. We're going to focus on insulin and fatty acid synthesis, or what we call the fed state. And so here's a metabolism overview. So we eat high energy nutrients, we digest them into monomers, and then the question is what happens. In some cases we store, we have excess. So excess carbon in the form of carbohydrates or proteins could lead to storage of proteins as well as fats, glycogen. And also we can burn these too. So we know that what we can do is the monomers can be oxidized into CO2. This is the most oxidized form of carbon. We make reducing power, which is converted into ATP during electron transport, as well as by substrate level phosphorylation. And this is done for growth, work, and maintenance. So the major metabolic hormones, there's a lot of hormones. So we're going to focus on 4 of them. We'll mention a few other ones as we go along. So insulin is released by beta cells in the pancreas. It's widespread, and insulin, they say, makes you fat. It promotes anabolism. So what it does is it takes the sugar in from the bloodstream into tissues, and those carbons are used to make fat, glycogen, as well as proteins. Opposite of insulin are glucagon, epinephrine, and cortisol. We won't focus too much on these in this particular lecture, but we'll see the fasted state next time. So what glucagon is the opposite of insulin. So it's made by pancreatic alpha cells, and it's only active in the liver. And what glucagon does is it promotes gluconeogenesis as well as carbohydrate release in the form of glycogenolysis. And so what we do is we increase the glycemia, we increase blood sugar levels. And there's also epinephrine and cortisol, which are also involved in increasing glycemia. There's other hormones too. So, you know, the idea of insulin and glucagon, this bimodal, biphasic, high sugar, low blood sugar state is a really good way to keep metabolism in your head. But there are things like leptin and neuropeptide Y. This is the more psychological and behavioral aspects of eating. So eating behavior is incredibly complex. And so leptin is a hormone made by the adipose tissue. And what it does, it's involved in feeding behavior because what it does is it actually inhibits the conversion of don't feed to feed, this psychological state of being hungry or not hungry. And so leptin mice, basically leptin deficient mice, I should say, are really, really fat. So leptin inhibits the urge to eat, and this is counterbalanced by neuropeptide Y. So there's a lot of hunger hormones. So insulin's a very famous molecule. It's incredibly important. Of course, we understand its role in diabetes. And so it's actually made as a precursor known as pre-proinsulin. And so what you see here is basically an alpha and beta chain separated by the C peptide. Notice that it has a number of disulfide bridges here. And these disulfide bridges are oxidized cysteine residues, and you normally find disulfide bridges on proteins that are secreted. And so as you might expect, insulin will be secreted, and it's modified by peptidases, and we get the mature form of insulin here, and there's something called C peptide, which might have a role in physiology. It's been studied for years. There's a couple of things about it. It's not tremendously important, certainly not at the level of what insulin is. So how is insulin released? Well, here we see a pancreatic beta cell, and we see glucose brought in by its transporters, and then it gets phosphorylated to make glucose 6-phosphate. We'll talk more about glucokinase and hexokinase in a moment. And then what happens is as it goes through metabolism, glycolysis, pyruvate dehydrogenase, TCA cycle, you generate more ATP. So you oxidize the carbons to make reducing power. The reducing power is converted into ATP. And it turns out that there's an ATP-sensitive potassium channel in the membrane of pancreatic beta cells. And it turns out also that ATP actually inhibits it. And when you inhibit the movement of potassium from the inside to the outside, you actually build up intracellular potassium, and that serves to depolarize the membrane. And when you depolarize the membrane, you open up voltage-sensitive calcium channels. The calcium rushes in, and the insulin that's stored in vesicles is actually released in a calcium-dependent manner. You should appreciate that the vast majority of things released from vesicles require calcium to do this. So what we see then is that ATP causes a depolarization of the pancreatic beta cell membrane, and that leads to insulin release through calcium-dependent processes. So you think of depolarization, you normally think of neuronal cells. So beta cells can be depolarized too. Well, it turns out that all cells can be depolarized, and this is basically movement of ions across the membrane. Now, the beta cells depolarize, but it doesn't give rise to an action potential like neuronal cells do. So in the case of a neuronal cell, you see depolarization here. Here's our -70 resting potential. And as you open sodium channels, what happens is that sodium rushes into the cell. This is analogous to the potassium building up inside of the cell. It becomes more positive inside of the cell. And then in the neuron, what happens is that the sodium closes, potassium opens, and then you repolarize, and you ultimately arrive back at the resting potential. So think of depolarization as the accumulation of positive charge inside of a cell. So what does the insulin do when it's released? Well, it goes to its receptors. And here's an example of the insulin receptor here. So here's insulin in the yellow. It binds to its receptors. And what happens upon ligand binding to the receptor, you induce a conformational change where you autophosphorylate tyrosine residues intracellularly. And what this does is initiates a signaling cascade. And in fact, insulin signaling is quite complex, but we've simplified it to think of glucose transport. So it causes the increase of glucose transporters on the surface of various tissues like skeletal muscle. It also promotes glycolysis, as we'll see, as well as protein synthesis. There's a number of other things it does here as well. For example, it promotes glycogen synthesis by expression of enzymes like glycogen synthase. We'll take a look at glycogen metabolism separately. So as I said, insulin promotes anabolism, and this is called the fed state. So what you see is that you take in dietary sugars, amino acids, as well as fats, and what you need to do is you put them into metabolic pathways. And so the sugars, like glucose, for example, will be phosphorylated, and you generate glucose 6-phosphate. Once you phosphorylate glucose, it can no longer leave the cell. And so what you have to do is you have to use it put it into metabolic pathways. And so you can make glycogen in the liver. You can actually do glycolysis, and then the formation of acetyl-CoA through pyruvate dehydrogenase. And I'll show you the details of fat and triglyceride synthesis in a moment. You can also use the Pentose phosphate shunt. All this stuff is on the AAMC MCAT Content Outline, which you can download in checklist format for free on our site. And so if you have an excess amount of glucose being consumed, then what happens is that you make fats and triglycerides, and then we're going to see that the triglycerides will be packaged into very low density lipoproteins, VLDLs. This is because they're very insoluble. In order to transport them to recipient tissues like adipose, what you have to do is solubilize them and have a way to move them around. Dietary fats that you consume, like through cheese, are actually made into chylomicrons, and the chylomicrons are generated in the intestine, and these blood lipoproteins will be transported to adipose tissue for storage as triglycerides. We'll see more about that in a minute. But so once you make glucose 6-phosphate, there's 3 states of glucose 6-phosphate that are heavily regulated. You can make glycogen by glycogen synthase, and as I mentioned, we'll take a look at glycogen synthesis as well as its degradation in a separate session. There's also glycolysis, which I think everyone is familiar with. We'll take a look at that as well today. And there's also the Pentose phosphate shunt. So glucose 6-phosphate has multiple cellular fates. And if we take a look at the Pentose phosphate shunt, what you do is you take glucose 6-phosphate and you oxidize it, and through its oxidation by glucose 6-phosphate dehydrogenase, you create NADPH reducing power. NADPH is used in several biochemical processes, including the synthesis of cholesterol and fatty acids. So, but one thing is that NADPH is not used in electron transport, it's only NADH. And so what you can do is create 6-phosphogluconate, which is an oxidized form of glucose 6-phosphate. You further oxidize it by releasing CO2, and you make ribulose 5-phosphate. This is a 5-carbon sugar. And then what can happen is that this can get integrated back into the glycolytic pathway, which only contains 3 and 6 carbon sugars. So one of the functions of the Pentose phosphate shunt is to integrate 5 carbon sugars as well as 7 and 4 carbon sugars into glycolysis. It's also designed to make NADPH reducing power, and you also make the precursors to generate nucleotides. This all occurs in the cytoplasm. So phosphorylating glucose occurs by the action of hexokinase or glucokinase. We saw in the pancreas that it expressed glucokinase. Glucokinase is also expressed in the liver, and it's quite important to distinguish where it's expressed in the liver versus say, the muscle. Hexokinase is in the muscle. So here's the reaction. We take glucose plus ATP, and we phosphorylate glucose, glucose 6-phosphate. No big deal, but we should recognize that once you're phosphorylated, you cannot leave the cell. It's trapped. And if we look at the kinetics of glucokinase versus hexokinase, so we plot out the velocity versus the substrate concentration, what we see are a couple interesting things. We see, first of all, that hexokinase saturates really fast, okay? And it has a very low Km. And we should appreciate Km is not only the concentration of substrate at 1/2 Vmax, but also a relative affinity constant. That is, the higher the Km, the lower the affinity. So if you take a look at hexokinase, has a lower Km than glucokinase. So the skeletal muscle isoform has a really high affinity, but it saturates super fast. And once it saturates, there's end product inhibition. That is, if you build up glucose 6-phosphate in a muscle cell, it'll prevent hexokinase from further catalyzing the conversion of glucose to glucose 6-phosphate. As a consequence, any new glucose that arrives in the skeletal muscle will not be phosphorylated, and it'll leave back into the bloodstream, circulate, and it'll end up in the liver. But notice that the liver enzyme has this really high Vmax. It's hard to saturate. That means that any excess glucose that the skeletal muscle doesn't want, the liver will take in. And as the liver takes it in, it'll ultimately convert it into fat. And that's how excess glucose is converted into fat. It's because of the nature of the varying Vmaxes and the Kms for hexokinase and glucokinase. And so the reason why cells have evolved different isoforms for metabolizing glucose, i.e. hexokinase versus glucokinase, speaks to the physiology of what's going on in the skeletal muscle versus the liver. The skeletal muscle doesn't want to make a lot of fat, the liver does. Pretty important to understand the distinction. So insulin has a big role in glycolysis. So what it does, it actually expedites the flux of carbon, oxidation of carbon from glucose into pyruvate. And so we've seen glucose to glucose 6-phosphate, you isomerize the fructose 6-phosphate, you phosphorylate again into fructose 1,6-biphosphate. And here's a really super important regulatory step. Phosphofructokinase 1, or PFK1, determines whether or not those carbons are going to enter into glycolysis or not. If you're upstream of this step, you can still go into glycogen metabolism, and you can still have the carbon go into the Pentose phosphate shunt. So it shouldn't surprise you that in the liver, as well as other tissues, that this enzyme is regulated. And the liver regulation is pretty important because it's regulated by insulin action. So over here on the right panel, what we see is that we have fructose 6-phosphate, and there's a substoichiometric amount that's converted into fructose 2,6-phosphate by the action of PFK2, which is only in the liver, and it's stimulated by insulin metabolism and signaling. So PFK2 will take some of the fructose 6-phosphate, convert it into fructose 2,6-phosphate, and then this is an allosteric activator of PFK1. And then what you can do is you can expedite the oxidation of the carbons in glycolysis. Insulin also stimulates the activity of pyruvate kinase, which is the last step in glycolysis. And so what insulin's doing is that it's involved in stimulating various aspects of the glycolytic pathway. And the idea is to pull carbon down as fast as it can into pyruvate. And what we also see is that if we're going to metabolize fructose, because we eat table sugar, which is going to be a molecule of glucose and fructose, what happens with fructose metabolism is that it uses a glucose transporter. But what you can see is that there's an enzyme known as aldolase that splits fructose 1-phosphate into glyceraldehyde and DHAP. There is no PFK step in fructose metabolism. And so what happens with fructose is that its oxidation via glycolysis actually bypasses insulin regulation. And there's a number of implications for this that people have noticed with respect to triglyceride and fatty acid metabolism. So fructose is not metabolized the same as glucose at all. And so what we see happen then is insulin stimulating glycolysis, whether it's going to be glucose or whether we have fructose coming into the system. And what we do then is we expedite the flux of carbons through glycolysis. We oxidize glucose into 2 moles of pyruvate per 1 glucose. Then you further oxidize those glucose, those what used to be glucose, into acetyl-CoA, which is converted, as we know, in citrate into TCA cycle. And here's the decision the cell makes when it's going to decide to make fat. If you have so much glucose coming down the pathway here via insulin stimulation that you overwhelm the TCA cycle's ability to oxidize citrate, the citrate accumulates. It's basically mass action. It's basically saturating enzymes by presenting it with so much substrate. Then what happens is that the citrate accumulates at a certain level, then it comes out into the cytoplasm, and the cytoplasmic citrate is going to be used as a substrate to make fat. And we'll elaborate on that by taking a look at the pathway of using citrate as a precursor for fat. So here's citrate up here. Notice that it's got 3 carboxyl groups. That's the origin of the name tricarboxylic acid cycle, TCA. And so citrate is exported from the mitochondria into the cytoplasm, and then a lyase enzyme splits it up into oxaloacetate and acetyl-CoA. Okay, so lyases are molecule or enzymes that actually split carbon-carbon bonds. And so then we see that the fate of the split citrate is to use acetyl-CoA as a substrate to make fats. So fatty acid synthesis is the opposite of fatty acid oxidation. Well, I'll show you beta reduction in a moment. And then the oxaloacetate is ultimately going to be converted back into pyruvate, which will be imported back into the mitochondria. There's a really important enzyme known as the malic enzyme here. It's a cytoplasmic enzyme that actually converts malate into pyruvate, but it generates the NADPH, and this NADPH plus the NADPH we saw made in the Pentose phosphate shunt are going to actually be used as energetic fuel to make fats through beta reduction. And so here's actually the beta reduction pathway. Here's the reaction. So the AAMC Content Outline specifically mentions the actual formulas and the yields for these processes. So what we're going to do is we're going to take 8 acetyl-CoAs, as you see up here in the equation, 8 acetyl-CoAs, and we're going to make a molecule of C16 with 0 double bonds. That's palmitic acid. That's the most common of the fatty acids. That's what fatty acid synthesis makes, synthase makes. And what we have then is we need 7 ATPs and we need 14 NADPHs. So it takes a lot of energy to make fat. So as you can imagine, the cells have probably thought about this and they're going to regulate it pretty carefully. So what we see is the rate limiting step is conducted by acetyl-CoA carboxylase. This biotin dependent enzyme takes acetyl-CoA that was derived from the pyruvate dehydrogenase complex, bicarbonate and ATP, and it actually makes malonyl-CoA, which is a 1, 2, 3 carbon derivative. So you add on a carbon. That's what biotin does, adds on carbons. This enzyme stimulated by insulin, it'll be inhibited by glucagon. And then anytime you see insulin activity upregulating something, you know glucagon will do the opposite. And here's how it works. The fatty acid synthase enzyme in the cytoplasm has two key cysteine residues. One's called the acyl carrying protein, one's called the condensing enzyme. And what you can see is that these cysteine residues act as nucleophiles, and they're going to attack, in this case, the malonyl-CoA. This is a thioester. And what you're going to see is a trans-thioesterification reaction. So what we're going to do now is we're going to look at fatty acid synthesis from the perspective of organic chemistry. And I would argue this is a pretty high yield way to look at organic chemistry because it has a biological relevance to an important pathway. So what we do is we attack the electrophilic carbon, make a tetrahedral intermediate, it collapses, and we release free coenzyme A. And an analogous reaction happens with condensing enzyme at the actual, with acetyl-CoA. So we do a trans-thioesterification. And so now acyl carrying protein and condensing enzyme are carrying the 2 carbons from acetyl-CoA as well as the 3 carbons from malonyl-CoA. And so let's take a look at what happens. So what you see here is acyl carrying protein and condensing enzyme are both loaded. And this acyl carrying protein entity actually decarboxylates its CO2 here, okay? And what you do is you generate a resonance stabilized carbanion here because these electrons can go into this and the O- can form. So basically it's resonance stabilized, so it's a really good nucleophile and it attacks the electrophilic center on the condensing enzyme. And what you end up doing is you end up transferring the 2 carbons of acetyl-CoA that were once on condensing enzyme over to acyl carrying protein. 1, 2, 3, 4. And now what we've done is we have 2 plus 2 is actually 4. But in order to do that, we had to lose a CO2. So it's actually 2 plus 3 minus 1 is 4. And so now we have the very nascent growing fatty acid chain. And you can continue this because what you can do then is you actually are going to reduce with the NADPH, this carbonyl, this ketone group here is going to be reduced to an alcohol. It's the beta carbon. So it's beta reduction. It's the opposite of beta oxidation. And then as we continue further, what you see happen is that you have an alcohol here, you lose water and make a double bond. That's a famous generation of an alkene through dehydration reaction here. And the double bond, as you might imagine, will be reduced to the saturated alkane form here. So this carbon that was once a carbonyl is now completely reduced using NADPH as well as dehydration. And then now what happens is that you have a translocation event. So you make basically you have this sulfhydryl will be a nucleophile attacking the electrophile, and you translocate. And then what you do then after you translocate, what do? You're going to bring another one in and you complete the cycle again. And what happens is that you go from 2 carbons to 4 carbons, you bring another one in, okay, and then you go from 4 to 6 to 8, all the way to C16, palmitic acid, using simple organic chemistry reactions that the fatty acid synthase, this multi-subunit enzyme that regulates fat synthesis. Now you've made a fat, but that's not a triglyceride. Now what you do is palmitic acid is the first one you make. Now you can make longer than 16 carbon fatty acids. The endoplasmic reticulum will contain enzymes for extension. It'll also contain various enzymes that can put double bonds in. So we'll just take a look at what we do with saturated palmitic acid. What you do then is you activate it, and you should appreciate that all fatty acids have to be activated with ATP, and you put on coenzyme A to make the thioester derivative, the CoA derivative. This thioester is actually a high energy bond. We often think of high energy bonds as phosphoanhydrides. That's true, but thioesters have almost the same amount of energy that's released upon hydrolysis that phosphoanhydride bonds do. So what we have then is an activated fatty acid, palmityl-CoA, and then we take glycerol 3-phosphate. Where does glycerol 3-phosphate come from? It comes from reduction of glyceraldehyde 3-phosphate in the glycolytic pathway. And so this alcohol is a nucleophile. So an alcohol attacks a thioester, and what you do is you esterify it, and you've actually added one of the fatty acid chains onto glycerol, but we know there's two other ones that we can add onto it to make the triglyceride, and the synthesis of the triglyceride occurs as follows. Here's actually our substrate molecule here. So we've added on one R. I've simplified palmitic acid by calling it R. Acyl transferase brings the second one on. Okay, so an acyl group is RC double bond O, where the R in this case will be 15 carbons, and you have your 16th for palmitic acid here. And so we add on, and now we have two R groups onto the actual glycerol entity. We dephosphorylate it by a phosphatase enzyme, okay? And so the substrate phosphatidic acid is converted into diacylglycerol. You should appreciate diacylglycerol as being a precursor for triglycerides, but also, in fact, equally important is that it's a signaling molecule for G protein cascades. So acyl transferase brings on the third R group, and now you've made your triglyceride. So you've made a triglyceride in the liver, but now we have to get it to where? The fat cells, the adipose. So let's take a look how that works. So here's the pathway: glucose to acetyl-CoA. Okay, amino acids can be converted into acetyl-CoA. Fatty acid to fatty acyl-CoA, and then the triglyceride. Here's the actual metabolic pathways which are being employed. We have glycolysis, pyruvate dehydrogenase, fatty acid synthesis, and in fact, we're going to see acetyl-CoA is a precursor for cholesterol, which is a precursor for steroids and vitamin D. So there's a number of important biochemical pathways which are used in the fed state, that is, insulin's promoting this. What we should appreciate then is that glycolysis, which we normally think of as a catabolic breakdown pathway, is actually used to make fatty acids. So anabolism and catabolism can be relative terms. And so here's what we do with our triglyceride. So if you make triglycerides in the liver from excess carbon, like you ate too much carbon in the form of protein, amino acids, sugars, etc. So what you do is you package it in the liver as a VLDL. But if you consumed preformed, exogenous fats, then what happens is you make chylomicrons in the small intestine. So chylomicrons and VLDLs are two lipoprotein particles that are used to transport the actual triglycerides to the adipose tissue. And so here's how it works. So as they circulate and they come to the adipose tissue, on the surface of the adipose tissue, there's an enzyme called lipoprotein lipase, or LPL. What lipoprotein lipase does, it actually hydrolyzes the triglycerides. And the reason why is because triglycerides cannot cross the cell membrane, but the fatty acids can. So in order to get a triglyceride from, say, the liver where it's made into the adipose tissue, you have to break it down and resynthesize it. And so LPL is the first step in doing that. So lipoprotein lipase is going to be hydrolyzing triglycerides, taking the fatty acids and bringing them across the membrane. And then the fatty acids plus glycerol 3-phosphate, which we know is made from the glycolytic cycle, is going to be used to repackage the triglyceride. And there you store fats in triglycerides, and they'll be mobilized by epinephrine or even glucagon in the liver. So here's what we have: glucose to acetyl-CoA, but now we're going to look at a separate pathway known as the mevalonic pathway, the mevalonic acid. And so this is a really important pathway because what we do is we're going to synthesize cholesterol. I think there's 14 Nobel Prizes that have been awarded for the synthesis of cholesterol. It's an incredibly important molecule. So what happens then is acetyl-CoA is converted into mevalonic acid. And then there's an enzyme that's inhibited by statins. This is called, these statins are used for cholesterol reduction. They're commonly prescribed drugs. And so when you're a doc one day, you're going to be prescribing statins to patients. What they do is they inhibit an enzyme known as HMG-CoA reductase. And what happens in that case, that you prevent the synthesis of all these downstream molecules. What are they? Well, they're called isoprenoids. So you have isopenyl pyrophosphate, dimethylallyl, geranyl, isopenyl. Right now, they kind of look like alphabet soup in a way, a lot of complicated molecules. But what they are is they're basically 1, 2, 3, 4, 5 carbon precursor molecules. And so just as amino acids are to proteins and nucleotides are to DNA, these isoprenoids or terpenes are to cholesterol and steroids. They're fundamental building blocks of nature. And without them, we wouldn't be living. And so what happens then is that cholesterol is normally synthesized by adding on multiple carbon units. And ultimately, what you make is a 30 carbon final structure that's chopped down to 27 carbons, and cholesterol is 27 carbons. So statins actually inhibit the synthesis of cholesterol. But you know, besides cholesterol, these molecules like geranyl pyrophosphate, these isoprenoids, are also utilized in reactions known as prenylation. What they do is they modify proteins. And when you modify them and stick all these hydrophobic groups onto them, you localize them to membranes. And so what we see here in content category 3A from the AAMC content outline is terpenes and terpenoids. Notice it's classified under lipids, fats, and it says steroids here as well. So it seems to me that the MCAT people want you to understand the mevalonic acid pathway. So with that in mind, let's do some really good passages which will challenge our knowledge, explore our boundaries of understanding fatty acid synthesis, fructose metabolism, as well as cholesterol. So let's get ready and apply our information to the MCAT, and let's get going.